Next, it's my pleasure to introduce my former graduate classmate and current colleague, Dr. Tim Guo, who today will be speaking about his work from his postdoc work at the Massachusetts Institute of Technology, MIT. And the talk is entitled Sense and Respond Payload Delivery Using a Synthetic Promoter in Engineered CAR T Cells. Tim, thank you for joining us, and please take it away.
Thank you very much, Payam. I appreciate the invitation. It's great to be able to attend another University of Toronto endeavor. And thank you for this opportunity again to share this work I did during my postdoc. So maybe following up on JC's initiation of this session, I'd like to talk a bit more about engineered T cells in the treatment of specifically immuno-oncology setting. So there's some work on engineering synthetic promoters to better regulate engineered T cell function. So as you may know, in the context of engineered T cells for adoptive T cell therapy, you take T cells from the peripheral blood, engineer them most commonly right now to redirect specificity against an antigen of interest through a T cell receptor or a synthetic molecule chimeric antigen receptor. And then you expand and reinfuse these cells back to the patients. In the context of engineered cells, you can think of it, breaking down into endogenous natural mechanisms versus synthetic ones, and the input and output you can get. So normally T cells receives an input through the T cell receptor, and that's how they recognize antigen. And the natural output they have is whatever endogenous effector responses they can elicit, for example, cytokine production, cytotoxicity, and proliferation. In the past decade or so, we've seen an advent of advances in CAR engineering, a chimeric antigen receptor, which now allows you to redirect the specificity and the effector function of T cells to a synthetic input in terms of any antigen that you can bind. Now, for us to really achieve the full potential of engineered cell therapy, we really need to fill this last quadrant down here, because we know that at least what we're doing right now with the synthetic input and endogenous output is often insufficient to achieve the required response in a clinical setting. So what we're hoping to accomplish is to, again, fill in this quadrant down here where we can have any input of choice, but have a synthetic output that can hopefully augment efficacy. The exact problem we're trying to work on here is in the context of solid tumors using CAR T cells, which again has been showing very little efficacy. Part of the problem there is the T cells are not sufficiently activated, either due to low antigen density or suppressive tumor microenvironment, which dampens the anti-tumor response. So you get not very much efficacy and obviously not much toxicity that goes along with that. So one way to improve the anti-tumor efficacy of these engineered cells is that you can equip them with additional payload, for example, inflammatory cytokines. The problem here is that when you constitutively produce these cytokines under a constitutive promoter, yes, you can drive anti-tumor responses, but at the time, it also unleashes off-tumor toxicities due to the systemic nature and the constitutive nature of this modality. So one way to selectively improve efficacy without creating too much toxicity is to be more selective in how you deliver these inflammatory payloads. And one way is that we can hijack the fact that these engineered T cells are already preferentially recognizing tumors through the engineered receptor. In our model, it will be a CAR, and take advantage of the signaling from the antigen receptor to drive the expression of a therapeutic payload. And hopefully that will redirect them towards more preferentially to anti-tumor response while minimizing off-tumor toxicities. So the point of engineering for us was really a synthetic promoter that can sense the antigen receptor signaling and respond by delivering a payload of interest. So give a bit more introduction on what goes into a synthetic promoter. Naturally, transcription regulation is downstream of a signaling pathway that activates a transcription factor, which binds to enhancer response elements that drives the expression of genes. And a synthetic promoter very much takes advantage of these natural components, where we have the response elements that binds to the transcription factor of interest, and this dictates what pathway is going to activate your promoter. Combine that with the minimum promoter that is required for the assembly of polymerase complexes, all of which is upstream of your gene of interest. So for this particular application, having an antigen-induced promoter, the field has been for a while using this conventional NFAT-based promoter, which is a transcription factor driven by T-cell receptor signaling. And the response element here is derived from human IL-2 gene, which again is activation dependent. So initially, this response element was used as a reporter to simply study T-cell receptor signaling. And a bit more recently, it's been used in a therapeutic setting to drive the inducible production of IL-12. And this was a trial done at NCI by putting these inducible vectors into tumor-infiltrating lymphocytes. So we were coming into this field and we were wondering, people have been using this NFAT for quite some time now, and without really looking at other alternative options, is there anything else that could be potentially better than NFAT? So our approach to identifying new synthetic antigen-inducible promoters was to look at what are known transcription factors that are either directly downstream of TCR signaling. So that's the MAP kinase pathways, NF-κB, and of course NFAT, which has already been studied, but also transcription factors that's secondarily upregulated by T-cell receptor activation, which can then also drive antigen-inducible responses. So we cloned several of these response elements, again with a minimum promoter and a fluorescent reporter, to test which ones can be antigen-inducible. Long story short, we narrowed down to 2 candidates that we want to study in comparison to the standard NFAT based on AEP1, which is a well-known transcription factor that's downstream of T-cell activation, as well as NR4A, which has not been really tested very much in the literature. For the first set of experiment I'll show you, we also included a constitutive expression module in the same vector, where a tag CD271 is expressed by a constitutive promoter to mark the engineered cells. So this way we can look at engineered cells and how they respond to activation based on this inducible fluorescent module. As the control, we have the tag alone. So we transduce this to primary human T cells and treat them with anti-CD3 stimulation. And we can see that the 3 response elements in different combinations of minimal promoters all behave similarly to the strong stimulation with anti-CD3. As internal positive control for T-cell activation, we also measured CD137 upregulation, which is a human T-cell activation marker. So now we have, especially in NR4A, a new antigen-inducible promoter that behaves, at least based on this assay, similarly well to the conventional NFAT. To study how these promoters respond when the signal is taken away, because for an inducible promoter, you don't only want them to be activated when they're signaled, but you also want the response to go away when the signal is taken away, so that it's tied to your pathways of interest. We switched the reporter to a destabilized fluorescent protein, such that the presence of fluorescent signal is more closely tied to transcriptional activation in this case. So very similar setup. In here, we also did repeated stimulation to measure how reversible these promoters are. As you can see, after 1, 2, or 3 repeated stimulations, all of these promoters, after stimulation, increasing signal, and very quickly within 1 day, largely returns down to baseline in terms of fluorescent response, showing that they are reversible. And also they demonstrated no toxicity, in that the frequency of the transduced cells, again measured by CD271 tag, remains fairly constant throughout culture after repeated stimulation. So finally, we move on into a antigen receptor setting. Here we used a mesothelin-specific chimeric antigen receptor. Mesothelin is a very common solid tumor antigen, and this CAR is being tested in the clinic for to treat various solid tumors. So a similar vector design where we have the constitutive expression of CAR as well as a tag to mark the engineered cells. And then upstream of that module, the inducible system, where we have different antigen response elements, along with the fluorescent reporter to mark antigen-induced response. We transduce them to primary T cells again, and here we co-culture with target cells that express various levels of the antigen. 293T is a negative control, and a lung and ovarian cancer cell line that express low or high levels of mesothelin, as shown below. So what is very interesting now is that we see when the CAR T cells are stimulated with the strong antigen of CAR8, they again respond very similarly like we saw with the anti-CD3 stimulation, where these engineered cells promoters have strong inducible responses. But when we stimulate with the weak stimulator having low levels of antigen, we saw that NR4A promoter responded significantly better than the conventional NFAT. And again, looking at CD137 upregulation, we know for a fact these T cells, when stimulated with A549, was less activated than the strong stimulator of CAR8, highlighting that there's a particular context where the NR4A promoter might actually be better than the conventional NFAT. Finally, in a therapeutic setting, we changed the payload to actual cytokines that we can study to help see how they help T cell response. So here we use the pro-proliferation cytokines IL-2 and 21 under the induced birth promoters and the same constitutive expression module downstream of that. Again, these cells, these constructs were delivered by lentivirus to primary T cells, and then we co-cultured the engineered cells with the same targets. And here we're measuring by CFSE dilution assay how much cells responded. You can see when there's no target, very little proliferation, strong proliferation when co-cultured with the OVCAR8 high antigen targets. And against these low antigen targets, A549, only the cells engineered with the NR4A promoter driving the inducible cytokine production was able to improve response compared to control being CAR alone. In fact, the NFAT was not able to improve proliferation in this low antigen setting. To confirm that this improvement was in fact payload dependent, we repeated experiments and compared the NR4A promoter driving the cytokines versus NR4A driving a negative control payload using the fluorescent reporter. And we show that it is in fact necessary to have the payload to have this improvement in response. It's not just some artifact we're seeing with having the synthetic promoter itself. So with those main points, I'll like to summarize for the sake of time. The system I've described here is a expression system where you can engineer primary human T cells, where you have the constitutive expression of CARs, but also inducible expression of payload, where the control of the payload delivery is now autonomously controlled based on the sensing of the antigen. And the key takeaway here is that we have identified a new antigen-induced promoter based on NR4A response elements, which seems to have better promoter responses compared to the conventional NFAT at levels where the immunogenicity is low, when it's weakly stimulatory. And that's very important because this is precisely the context where you need to deliver payload. It is under these weak stimulatory contexts you need to enhance response, and that can see the true benefit of an inflammatory payload. And you can envision beyond just cytokines and other cell extrinsic payloads, and that can be used to modify the cells themselves or neighboring cells. You can also use them to deliver cell intrinsic payload to, in a context-dependent manner, reprogram the cells as necessary by expressing, for example, other transcription factors or microRNAs or cell surface receptors. So with that, I'd like to stop there and happy to take any questions.
Tim, that was a great talk. I'm going to jump in here and ask a question right off the bat. Thank you very much for that. I mean, this is a really exciting strategy of delivering a payload. I'm just wondering if you envisage that you can combine that strategy with what JC was just talking about earlier. And I'm thinking actually very much in the context of some of these new vaccines that are being developed. If this is a good strategy to combine everything and actually use it in a vaccine strategy. So what are your thoughts?
Vaccine, just to clarify the question, Eleanor, so vaccine strategy is in delivering the antigen in a context-dependent manner?
Or if you know
enhance the response.
Or even ahead of time, if you know the T cell, specific antigen, just develop that with JC's strategy and then use your payload. Some sort of a combination there.
Right. So I think that's the exciting part about cell engineering and some of these advances in synthetic biology. We're really no longer limited by what nature has provided for us. We can really start to think about not just what we have been studying, but unleash our full imagination and what we need to do. And yeah, absolutely. The beauty here is that this is a platform, and in theory, any gene could go in here. Of course, this is limited by your gene editing capacity, right? But like a lot of these strategies are nowadays, but in fact, whatever you want to put in here can really determine what kind of response you get. It could be immunosuppressive to treat autoimmune disease, right? So no, absolutely, I think there's a lot of context where this could be applied.
That's really great. Maybe just one or two quick questions from the audience team. One quick question is if you have any comments on how different promoters may be intrinsically, differentially, regulated by mechanisms in the cell that would affect the stability of the expression of whatever you're trying to modulate.
I think that is the point, right? I think that is the reason why we need to try out different response elements. NFAT, NR4A, and many other transcription factors, they're all activated by T cell receptor signaling. But clearly there is some functional differences, right, between these transcription factor driven response elements. So there's definitely something that we probably don't fully understand yet, and which is why we need to empirically test different options, even though they're under the same signaling pathway of interest.
That's a really great point and a great talk again. We have two more questions in the Q&A section that I encourage you to take the time to answer. One is about bystander effects of CAR T cell therapy, like unregulated cytokine induction, and the other is to comment on using this sort of technology to treat something like long COVID. So any quick thoughts would be appreciated, and then we can move on to the next speaker.
My pleasure. I will answer in the chat.
Thank you so much.
Yeah.